Okay, so today we are going to talk about chapter 4 of your book on feathers and molt. We're primarily going to focus on four things today. We're mostly not focus on molt. We'll talk a little bit about molt, but mostly we're going to talk about major feather regions, um, feather development, feather structure, and types of feathers. Oh, let's see here. There we go. So these first diagrams are not in your book. A lot of the diagrams that I'm providing today aren't the same diagrams as diagrams in your book, so I've provided a mixture of things that are from the book and from other texts, and all of those texts are cited on the first slide there that we just looked at. And this one comes from Peter Pyle's Guide to the Birds of North America, which is um, well, it's actually sitting right here beside me. Identification Guide to the Birds of North America. It is used by field ornithologists like myself. It describes in great detail molt strategies for different species of birds, which is really, really useful for aging them. Um, and it's just generally a really wonderful book if you're interested in some of the more niche knowledge of avian biology. So, a useful thing to understand is just how a bird is sort of put together. So, and I mean in terms of just its exterior, most superficial form, and that is the feathers, right? When you look at a bird, what you see is almost entirely feathers. If you take the feathers off of a bird, if you think about looking at like a chicken that's been plucked, it looks completely different from the animal you see with feathers. Kind of like if you shave your cat, they suddenly look very skinny. When they have feathers, they look full and robust. The same thing is true of birds. And so birds are covered in contour feathers that give them their shape and also create a sort of aerodynamic fuselage for flight. And beneath those contour feathers, there are other feathers, and we're going to take a look at those different feathers toward the end of this lecture. But right now, what we're looking at in these diagrams are the regions of contour feathers on the bird. Most of these regions are really just labeled in a very straightforward way that is not difficult to understand. So if we look at this diagram on the left here, things like the chin, the throat, the breast, these are feather regions that these labels are referring to, but they're just named for the general region of the body that we're already familiar with. So I'm not going to go over all of those, but you should familiarize yourself with them. They are fair game for an exam. One label that's not shown on this particular diagram that I wanted to add was the flank. So the flank is the side of the bird. A lot of birds, if you look at them, particularly, I work a lot with passerines, so this is sort of the context that I'm coming from. Particularly in small passerines, you will see a different color on the flank or a part of the flank than you will down on the belly of the bird, and that's a useful, sometimes identification characteristic. Another thing I want to point out on here I think is really important is the structure of the wing and tail feathers, the flight feathers. The flight feathers are themselves a type of contour feather. They're a type of contour feather. So these are contour feathers, all these body feathers, but also the flight feathers themselves, right? They're the superficial feathers that give the shape and aerodynamic structure of the bird. But the flight feathers obviously are a little bit different because they have a function toward producing lift and reducing drag and things like that. They have a direct function toward flight. So the tail feathers are called rectrices, and the major flight feathers, which is this row of feathers right here on the wing, are called remiges. So you have rectrices and remiges. The feathers of the tail and the feathers, the flight feathers of the wing, these feathers are counted from the innermost to the outermost. So you go to the inside and you count. So here that's Rectrix 1 and Rectrix 1, Rectrix 2 and Rectrix 2, etc. You count outward. And this has to do with the way the sort of what we call like the standard version of molting, the standard molting pattern. And that is birds replace feathers from the inside out. So we count the feathers in the same order they tend to be replaced. That being said, there are many molting strategies in birds, both in terms of seasonally when birds molt and in terms of the patterns of molt. So, in terms of the patterns of molt, some birds do the complete and total opposite of this. They molt from the outside in. It's just not the more typical molting pattern. In terms of seasonally, most birds, at least in North America, seasonally molt or replace all their feathers once in at the end of the breeding season, typically. This may happen at a migration stop off. It may happen right at the place they're breeding. It really depends on the species, and there's quite a bit of variability in that. And for some birds, that's the only molt they'll do every year. They molt once at the end of the breeding season, and then next year at the end of the breeding season, they replace their feathers again. But other birds will molt twice a year. They'll molt once at the breeding season and at the end of the breeding season, and then once before the breeding season. The birds that do this are birds that have two different plumages, typically a breeding plumage and a non-breeding plumage. So there's variability both in molt in terms of the molt pattern and in seasonal strategies. Now, when we get that's passerines, that's North American passerines I'm talking about. Molt strategies vary greatly in different groups of birds. The molt of woodpeckers, for example, or the molt of owls is quite different in terms of molt patterns and seasonal strategies, even from molt in small passerines. There are a lot of differences in different groups of birds. But let's get back to orienting ourselves to this bird here. So we've got our rectrices. We talked about the rectrices and how they're counted. Then we have our remiges. So the remiges aren't quite as simple as the rectrices, right? And just to point out what I'm talking about here, these tertials, secondaries, and primaries, these feathers here, are the remiges. The tertials, typically, they're not doing much to generate lift. The tertials sit close to the body, and actually when the wing is closed, they protect the other flight feathers. With the secondaries and primaries are important for flight and lift generation, particularly. And the reason that we don't just have, like in the rectrices, just call them all, like, why don't we just call these all, remiges, remige 1, remige 2, remige 3, remige 1, remige 2, remige 3. We could count them like that, just the same way we did the rectrices. Instead, we have primaries, and then secondaries, and then tertials. What makes them different? That's the question that we're asking here. Well, I'm going to use my arm as an example. The bone structure of a bird is similar to ours in terms of the forelimb here. We have a humerus, radius, and ulna, and then the bones of the hand, which is called the manus. They have the same thing. The bones of the manus are a little bit different. Particularly here in the wrist joint, there's some differences, but the structure here is the same. Now, if you were to look at where the different feather groups here on these remiges attach, you would see that they're directly associated with different regions of the forelimb. The tertials attach either right at the joint of what would be my elbow, or attach back onto the humerus itself. The secondaries attach to the forearm, or the radius and ulna. So that's the region of attachment. And then the primaries attach to the manus, to the bird's hand. We will take a look at that skeletal structure more closely later. In some birds, the attachment is directly to the bone. In different in these different groups of feathers, and in some birds, it's associated with that region of the bone, or of the skeletal structure, I think is probably a better way to say that. So that's the remiges, and that's why you have primaries. The primaries are attached to the manus, the secondaries are attached to the ulna, or associated with the ulna, and then the tertials are associated with, again, the alacranon process, I believe is what it's called, of that where the ulna comes down and meets with the humerus, um, and then the tertials are attached so that to that process, or the humerus itself. And that's the remiges. So now you understand retrices, you understand remiges. Let's talk about these other tracts of feathers up here. Above the flight feathers, you have a bunch of coverts, and you'll see the same thing is true for the retrices as is for the remiges. You have coverts that lay above them. So here we have the upper tail coverts right above the retrices. They stick out, they're feathers that stick out over the base of the retrices. Here we have a series of primary coverts that sit over the primary remiges, and we have a series of greater coverts that sit over the secondary remiges. Note they are not called secondary coverts. That's not very intuitive, but there's a reason for that. This entire series of coverts over here, the greater, median, and lesser coverts, all together are called the secondary coverts. So you have secondary remiges, secondary coverts. Secondary coverts include the greater, median, and lesser coverts. And then for primary remiges, you have primary coverts. You also have up here, really doesn't show here, it's 2 to 3 little feathers here that are called the alula. And these, when we get to talking about flight control, are a really important part of the wing. These are a couple little feathers that most people wouldn't even think about existing, or are certainly being important, but they actually have some significant contribution to flight control. If we look at the face of a bird, which I don't want to spend a lot of time on here, you'll see that there are various regions that we can refer to on the face of a bird that are feather regions that can be useful in identification, and so they're worth learning about. Here in this example, some things of particular note are, I think, the crown, supercilium, and the auricle. The auricle are the feathers around the ear hole, and we'll get to looking at the ear structure and how birds hear when we get to that that chapter. The supercilium is just sometimes just referred to as like a stripe. It's just a line of feathers running up above the eye, and is useful in identification. But all of these structures are useful in identification. Okay. Excuse me. So pterolysis, I just had like a stroke there. Pterolysis is the pattern of feather growth on the bird. It's actually the pattern of feather follicle distribution. And so feather follicles in many species of birds grow in these sort of lines or regions, and then they don't grow in other parts of the bird. This is really easy to see. This is a picture from your book. If you were to look at a plucked chicken or a plucked turkey, you will see regions that are bumpy, and they tend to be in these rows, and then regions that are more naked, like here and here. Where there are no bumps, there are no feather follicles, which means that is a place that's going to be always naked on the bird because the follicle is where the feather arises from. So pterolysis is just a description of the pattern of these follicles as they occur across the body of the bird. Each tract is a pterale. So there's different tracts on here, and each one is a pterale. So it's a region of feather growth. The regions that do not have feathers are called apteria, which is just lacking no pterale. They have no pterale, so that's what that means. So you've got regions that grow feathers and regions that don't grow feathers. The regions that don't grow feathers are, of course, naked. Now, you might go, well, that doesn't make any sense. I've seen lots of birds, and I don't really think I've ever seen a naked bird. And you would be right, because the way feathers grow, they grow from the pterale, these regions, and they cover the naked regions of the bird. But you can see these naked regions on the bird pretty clearly if you hold a bird in your hand and you blow into the feathers with a straw. There are regions you'll see the feathers, they'll spread from the pressure of the blowing through the straw, right? The feathers will spread open. And you can see the naked regions, the apteria, on the bird. There are some birds, some of the ratites, and in particular penguins, that don't have any really strong patterns of pterolysis. They don't have like tight regions of pterale and apteria. They're just covered kind of uniformly in feather follicles. And particularly for penguins, this should make a lot of sense. Penguins have to keep water from getting to their skin. They're swimming around in ice cold water. They have to have a shield that is extremely uniform and covering their body to protect them from being exposed directly to that cold moisture. They can't have the water getting through their feathers. The diagram here is just showing some of these tracts. I will tell you right now, I'm not going to ask you to memorize these tracts. I would point out on this diagram an important structure in maintaining feathers, and that's the uropygial gland, which you see. And in this uropygial gland is right on the tip of the hind end of the bird. Okay, and it produces oils that the bird uses to preen with. You will see this behavior in birds if you watch them, where they reach back with their beaks and they it looks like they're, I want to say, nibbling their butts. They're chewing back on their back ends. What they're doing is they're getting those oils from the uropygial gland on their beak, and then they can spread those to their feathers. So you'll see them go nibble their bums, and then they turn around, and then they might run some of their feathers through their beak. They're preening, they're cleaning and maintaining the condition of their feathers. Feathers are very important. Like I said earlier, they're only molted in a lot of birds once a year. Some birds, of course, like I said, more frequently. But that means they need to be kept in good condition until they can be replaced again. And this is a big part of keeping them in good condition. Okay, so that's the overall picture of where feathers are on birds and major feather regions. Now we're going to take a look at the more zoomed in structure of a feather and how even a whole wing works. First, the feather has a feather shaft. And that feather shaft is this entire central region of the feather that's kind of more rigid. The shaft itself can be broken into two major regions, a calamus and a rachis. The calamus is sort of the naked part of the feather, and the rachis is the part of the feather that is surrounded on either side by a vein. So, right, this is a vein, and this is a vein of this feather. Veins themselves may be plumulaceous or pennaceous, or some combination of plumulaceous and pennaceous. These veins are made of things called barbs and barbules that we'll take a look at in a minute. And the barbs and barbule structure of the pennaceous parts of veins, or whole pennaceous veins, form an interlocking meshwork that holds them all together and makes a rigid structure. You will see if you pick up a feather, like a turkey feather or a feather at the zoo, um, and you take the pennaceous portion and you pull on it gently, you'll see it sort of unzips, and that's because it is essentially velcroed together. The very fine microstructure of the barbs and barbules, it acts a lot like velcro to hold the whole thing together. And you'll also see if you take the feather and you grab it and you pull the barbs apart, if you take it and you do this and slide your fingers over it on both sides of the vein, you can actually zip the feather back up. uh, so it's all held together in this fairly static structure. The plumulaceous portion is made up of barbs that don't have the intricate interlocking functions uh, that the barbs of the pennaceous portion have. They're more just like long wispy chains. Um, they have little bumps on them called nodes or nodules, but that's not particularly important to this discussion. So this is the main parts of the feather. If I refer to distal on a feather, I'm referring to toward the tip of the feather, and if I refer to basal, I'm referring to toward the base of the feather, where the feather attached to the body of the bird at the calamus, the naked part of the feather. So we can look at this in a little bit of a different way. These diagrams come from a really awesome book. Do I have it sitting here? I do. It's called the Manual of Ornithology. It's cited at the beginning of this lecture. And so we looked at a lot of this stuff, right? You see the shaft, the rachis, the calamus, the two veins that are pennaceous and plumulaceous in nature. Um, but then we have some other stuff. For one, we see here a zoomed in cross-sectional detail of the rachis, and we see the rachis is not round. And this is true if you look at a turkey feather, for example, which is a very large feather. You can see this very easily. If you look at the actual structure of a turkey feather, you will see that it's got a groove in it. So it's shaped a little more like an I-beam, like that would be used in construction of buildings. And this gives this rachis incredible strength and resistance against the pressures of wind on the feather. It helps keep the feather from deforming. It is a very strong structure. Some other things that we see that are different. We see here labeled a superior and inferior umbilicus, and it's really easy to see the inferior umbilicus on a large feather like a turkey feather. Just pick it up, look at the bottom of the calamus, and what you will see is a hole. There's a very clear opening in the bottom of that feather. That opening is the inferior umbilicus, and what that is it's where an artery, an axial artery was running into the base of the feather during its development. You can sometimes see retained bits of pulp tissue called pulp caps in here, and they just look like little kind of usually whitish colored lines in the calamus. And we'll talk about the pulp tissue here in a little bit. Um, other things to talk about on this. So we talked about the pulp caps a little bit. I think that covers pretty much the more zoomed out detail of a feather pretty well. So let's zoom in now. If we were to look at a feather and we look at the rachis, we would see that coming off the rachis, the things that make up the veins coming off that rachis are these fairly complex structures uh, called barbs. And barbs look a little bit like Christmas trees. They have a long central shaft themselves. That's called the ramus, which you see here. And then from the ramus, there are these things called barbules. And so altogether, the combination of the ramus and the barbules make the barbs. The ramus is quite interesting. Um, and this really right here, this should probably just for clarity say not barbs but rami, plural of ramus. They're not they're not round either. Just like the rachis of the main feather shaft is not round, these aren't round either. They have an interesting sort of comma shaped structure. I don't know why it says comma equal shaped here. That should just be comma dash shaped. But inside of this structure of the ramus of every barb is a pith and cortex, and that's actually true of the rachis as well. So it's a sort of spongy tissue. It gives some flexibility to this structure and also lightens the structure. One of the reasons the feathers are so very light is even within their most tiny interior structures, you have some hollowness to those structures. But this comma shape is going to be very, very important for maintaining the overall wing structure. And that might seem hard to imagine because we've zoomed in. Right now we're incredibly zoomed in on the feather, right? We're not just looking at like one vein. We're looking at one barb in one vein, of which there are a tremendous number of these barbs in a single vein. Um, but it's these structures here, the barbs, that are going to hold whole wings together. So the structure is very important. The barbules coming off this, we have a distal and proximal barbule. That is a barbule that points toward the tip of the feather and a barbule that points toward the base, toward the calamus of the feather. And there are many, many, many of these barbules. At the ends of these barbules, we have two different structures, and it's important to understand both of them. So on the proximal barbule, we'll start there, we have a dorsal flange. And so what this is it's just a little hooked lip on the proximal barbule. It's just rounded. That's what the dorsal flange is. And then the distal barbules have what are called barbicils or hooklets. They're both perfectly appropriate terms for this. And that's what you see over here. What happens is if you line all these barbs up, right? So you've got these rami running one way, let's just say running straight into the screen here as it kind of is in the diagram, and then you've got poking in one direction, you've got flanged extensions, barbules, and on the other direction you've got hooked extensions, barbules. The hooks on one barb reach up and grab the flanges on adjacent barbules. So you've got hooked barbules, the hooked distal barbules grabbing the flanges of adjacent proximal barbules. This is that when I said you can unzip and zip a feather up, this is what causes that. When you take pressure and you slide a vein together, what you are doing is you are putting hooklets of one barb in contact with flanges of another barb so that they latch back a hold of each other. That's all you're doing. This is the sort of Velcro system that keeps veins of feathers together. Now that's difficult to visualize, so we'll take, I really like this diagram from your book, which I think helps to sort this all out. If we zoom in on this feather, you can see here's the rachis of the feather. That is uh, the veined part of the feather shaft. And coming off of that are the barbs. Okay, and then the barbs themselves, this part of the barb is called the ramus, right? Have a ramus and barbules sticking off of them. And we have proximal barbules. These are the barbules that are more or less pointed toward the base of this feather. And then distal barbules, the ones pointing toward the tip of this feather. Here the distal barbules are being indicated by the darker green and the proximal by the lighter green. And what you see is when you take these two barbs and you set them close together, as they overlap, the hooklets of the distal barbules reach up and hook into the flanges of the proximal barbules. And this is what this looks like, just zoomed in on two barbs. Here the hooklets have grabbed these flanges. That's how feathers stay zipped together. It's a rather complex structure. That's why I said they kind of look a little bit like Christmas trees when you zoom in this far. So that's how feathers stay together. Another question might be, if we think back to our wing diagram we were looking at earlier, and we have the primaries and the secondaries, those flight feathers. Those flight feathers have to stay together too. Like each feather itself has to stay connected to another feather in flight. And they're not attached to each other in a physical sense, right? They are technically free to move around wherever they want to go. And you would think if if a bird is flying into, say, a gust of wind, maybe it would separate those feathers. That would be bad because the airfoil structure that gives birds lift would be disrupted and the bird would fall out of the sky. So the question then is, how do the feathers themselves not just get blown apart from each other? This is done with something called lobate cilium. So let's take a look at this is two feathers. This is primary 5 and primary 6. I think this, if I remember correctly, this is from a pigeon in this particular paper from Matloff et al. 2020. And we're going to take a look here at this M diagram, right? We're zoomed in on this surface, and what we see here is P5 is the feather on the right here that we're zoomed in on, and P6 is the feather on the left. And we see the zone of overlap where these barbs are reaching out and laying, P5 is laying over top of P6. If we tilt this and we look now, instead of at the surface, but look in cross section, we can see P5 here laying over top of P6. So these are cross sections of the barbs of P6, and these are cross sections of the barbs of P5. And what you see is there are little, what they're called lobate cilia that stick up, and what they are doing, and it's very hard to actually tell this from this diagram, but what they do is they fit into a sort of groove at the bottom of the rami of the barbs, right? So this is a ramus, that's a ramus, that's a ramus. This is a ramus, this is a ramus, this is a ramus, and here where the barbules are sticking out, there are lobate cilia on the distal barbules primarily, and they stick up and grab a hold of the rami of the barbs on the feather that's sitting on top of them. So so this is like a bed of hairs that reach up and grab a hold of these barbs. Diagrammatically, if we look at this, it's maybe easier to understand because we have here the ramus of a barb. We have this distal barbule sticking out, and it's got these lobate cilia like you see here. This is a lobate cilium, that's just the singular of cilia. And it's basically a hair-like structure. And what you see here is this is the feather above. This is a ramus of a barb, and it is being grabbed by this lobate cilium. So what that means is the feathers themselves are held together. Like that is the singular feather is held together by a very simple Velcro-like mechanism of hooks and flanges, and then feathers are held to other feathers by a not dissimilar mechanism of little hair-like projections grabbing a hold of the rami of the barbs of overlying feathers. And so everything is just uh, structurally grabbing at everything else in the feathers. But this is very important. This is how the flight feathers actually stay connected to each other in flight. The asymmetry of flight feather veins is actually indicative of some of this function. So they're asymmetrical because that is each feather is independently its own little tiny airfoil, for one. Um, but more so, an important aspect of this is that the rear edge, oh, I gotta go to the screen, the rear edge of the feather here has regions of more of these lobate cilia, which means they're particularly good at grabbing a hold of adjacent feathers in those areas, and so that tends to be the rear edge of each of these feathers. That's good at doing that. So when you look at a flight feather and you're looking at the relative difference of the two veins, one vein is fairly thin and narrow, and the other vein, the 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 rear edge of that feather is wider. That wider edge will also have more and larger cilia to help grab a hold of uh, adjacent feathers. The vein itself, the the leading vein of a flight feather, will often be what is called a marginate, and you can see that here. This is like a place where someone has looks like they've notched this feather out. There are different hypotheses for why this is the case. One of those is that um, you create, when you do this, the ability for these feathers to act as individual airfoils. This is particularly true of flight feathers that are primary flight feathers toward the tip of the wing. So like primaries like 9 and 8, 7, those outer primaries. And if each one of them acts as its own airfoil, it theoretically reduces drag. And you will notice in some gliding birds, like hawks, if you watch hawks fly, you'll see the tips of the wings. Those feathers are not connected to one another; they're separated. So you'll see um, gaps when they're in flight in those feathers. And the idea is what actually happens is because they're their own little tiny airfoils, they reduce drag at the wingtips, and then that increases glide efficiency. That's one hypothesis for emargination. Um, Another hypothesis is that by having these emarginate tips and unlinking the tips of those outer primary flight feathers, the tips can bend. They can bend and rotate freely, and this may increase, or I should say reduce, issues with wingtip stall. Um, and that is something that can essentially be think of as like a micro adjustment to the angle of attack in flight. Now, we haven't talked about how birds fly yet, So if you don't understand angle of attack, that's okay. The third hypothesis is that it may be an adaptation for vertical takeoff because it overall reduces power requirements in vertical takeoff. And there is some pretty interesting evidence for this. There is a paper cited at the beginning of on the first slide of this lecture that is worth looking at because it investigates the presence of emarginated feathers in different groups of birds, and it comes to the conclusion that many gliding groups of birds don't have these emarginate primary flight feathers, and so it's probably not an evolutionary adaptation for gliding birds specifically. I think there are arguments to be made against that, but it is a really intriguing paper that looks at different groups of birds and the feather morphology and comes to an interesting conclusion about feather evolution. So it's worth reading. Here we have now, so we should have a pretty decent understanding, I think, now of where feathers are on birds and what structures of feathers kind of look like at the micro scale, and then how those structures hold feathers together and entire regions of flight feathers together. What we need to understand now is how does a feather develop at all? And so feathers develop again from feather follicles. And a feather follicle, what it is there's a little region of thickened epidermal tissue, and the dermis beneath forces its way up into that region of thickened epidermis. So the dermis pushes up into the epidermis. That's what you see in figure A here. This is the dermis pushing its way up into the epidermis, creating a little nubbin, which we might call a papilla. What happens once the dermis pushes its way up into the epidermis responds sort of by on the edges, in this what they're calling the zone of growth here, pushing its way down into the dermis. So the epidermis is pushing its way down, and what it does is it creates a fold on either side of the base of this papilla that's elongating into this feather follicle. The innermost layer of the epidermis here is going to become what's called the epidermal collar, and it is where feather development is really going to be taking place. The majority of the interesting stuff that we're going to talk about is occurring in these particular epidermal cells. And in fact, we're going to get these things called keratinocytes, and those cells are going to make keratin, and that is what is ultimately going to give rise to the feather. A lot of the development of keratinocytes. But note that this creates a double-walled structure. If we take a slightly more zoomed in look at that, this is the same thing I was talking about earlier. Here's our slightly thicker layer of epidermis. Here the dermis has intruded upon the epidermis, and the epidermis has intruded upon the dermis to create this fold, and ultimately you end up with this double layer of epidermis. Some things you might notice here that are kind of interesting in this picture in particular. One, this double layer. In this picture, you've got this big gap it's showing, but it's not a huge gap like that. It's a tiny, tiny gap. It's basically cells shoved right up against one another. In this double layer, you have the connection to the rest of the skin, which is the outer layer of the epidermis, and then you have the epidermal collar, which is going to make the feather. But one might ask then, well, what is the point of the dermis in all of this? What's the dermis doing? Well, the dermis creates a pulp tissue, and that pulp tissue is highly vascularized. And ultimately, the dermis creates growth factors and supplies the blood supply, the nutrient supply to the feather so that it may grow. It's how we harvest all of the materials we need to build the structure that we're going to build above the pulp. And that's the feather itself. And so right here, where the axial artery is coming in, this will, in the final feather, when you pick a feather up off the ground, that's where the inferior umbilicus will be. It's going to be right where this axial artery is entering. Note that there are muscles attached here. Most feathers, but not all feathers, have a very minor little muscle attachments. This is, as an example, how birds can fluff out their feathers because they have little tiny muscles attached to those feathers. This pulp and the artery and everything else in here that's dermal will almost all be reabsorbed and cut off from the feather once the feather is done growing. So this is a temporary structure. And we'll come back to some importance of that when working with birds here in a few minutes. Let's take a look at the epidermal collar here. So the epidermal collar has a dorsal and ventral region. Keep in mind, well, I'll just start here. If we just imagine that there's a circle and it's got a dorsal region and a ventral region, and the ventral region has a breaking point, and the dorsal region is where the rachis of the main feather shaft is going to develop. I would like you to keep in mind that the breaking point, the point at which the feather will open here, is not directly across from or directly in opposition of the rachis. The breaking point here for the dermal collar is actually going to be off center on some feathers, and that's because what's going to happen here is this ring is going to, this is a cross section, but this ring is going to open up and make the feather. And this is going to be I need to look at this, I'm sorry. This ring is going to open up and make the feather. This is going to be one feather vein, and this is going to be the other feather vein. Some feather veins are asymmetrical, right? We have got a narrow vein on a leading edge of a flight feather and a larger vein behind it. That's why this ring is not a perfect ring shape. This is a larger vein than this. Okay. So this is happening at this collar, right? And so you have to imagine a sort of tube, and the feather's growing in this tube. That's why it's a ring, because the feather right now is growing in a tube. The tube will eventually, if this is a tube, it will actually break open this way. It'll break open like this, where the rachis is here, and so the underside of the feather is the inside of this ring, and this is the upper. This is the ventral surface of the feather, and this is the dorsal surface of the feather. Hopefully that makes sense. If you can just imagine a straw with a break in it, and you take the straw and you split the straw at the break, what is facing down, once you do that, this surface here, is the underside of the feather, and what's facing up is the dorsal side of the feather. Okay. Within the epidermal collar, in this cross section, you can see the barbs growing, and you see the barbs, maybe very unintuitively, are the way the barb ridges are growing. They're kind of like really far out away from this rachis. Well, how the heck is a barb ridge out here attaching to this rachis? And that has a lot to do with how these barbs actually grow and attach to the rachis. They don't grow from the rachis. This kind of is depicting what's happening. Um, they grow uh, at the epidermal collar. They're initiated within the epidermal collar, the growth is, and then they migrate. The bases of each one of these barb ridges migrate towards the rachis as the rachis elongates. So as the rachis is getting longer and longer, the uh, the barb uh, the barbs are migrating to the rachis and attached to the rachis in that manner. So there's migration of these cells toward the rachis at the same time again that the rachis is elongating. This diagram is either very, very enlightening in how this works, or it's completely confusing, depending on how closely you read the text. But this is the placode. This is the beginning of the feather follicle, is what it is. And once the feather follicle forms, um, we have our epidermal collar, and the epidermal collar will initiate the growth of the barb ridges and of the initial feather shaft rachis. These barb ridges will grow down to the collar and they will begin to migrate toward the base of the growing rachis. And so that's what this is showing here. This migrate. See how these are barbs. Technically, they're called barb ridges at this point, but they're barbs. They're rami. And what you see is they look sort of twisted. And what's happening is the base of these rami is moving, and you can follow the arrows. It's moving in this direction, like a conveyor belt, toward the rachis on the dorsal side of this structure. At the same time, new barbs have to be formed on the ventral side of the epidermal collar. So we've got, just to orient you here, this is the dorsal side of the epidermal collar where the rachis is going to grow and the first barb ridges are going to form. As these elongate and reach the bottom of the epidermal collar, they migrate toward the base of the rachis and fuse into the rachis. And new barb ridges are formed in the epidermal collar on the ventral side of this structure, the feather shaft. That's a bad word to use for that because it's a little confusing. Within the epidermal collar is a more appropriate way to say that. This is just showing this growth of the ridges, how the barb ridges grow and are pulled around the epidermal collar to fuse with the rachis. And I think this picture is one that if you can understand this, I think it makes a lot of sense how this process works. Note that these barb ridges up here are fused to the rachis, right? This long stalk here is the rachis. And see how this one and this one aren't, but see how they're bent in the direction of the rachis. What will happen is as this rachis continues to grow and new space is available for attachment, this and these two barb ridges will be pulled in and fused to the rachis. What that will ultimately look like, of course, is a feather that's all rolled up. That's what you see here. And the sheath is something we haven't really talked about yet, but the sheath that's surrounding the feather will eventually degrade. So the whole entire feather in this process is covered in a thin keratin sheath, as sort of a protective structure that's there while the feather develops. Once the feather is fully grown, though, the sheath degenerates. And when it does, you've got that straw-like structure. You've got a tube of right, barbs on a rachis, and it opens up like this. And that's what you see here. That's the unfurling of the feathers. So that is how a feather develops. This is when this happens and you see it on an actual bird, it's pretty cool. This is a bird we called, I think this was Big Fluff. This was a chicken I hatched a couple years ago in one of my classes, and we were looking at the feather development a little bit in that class. And you can see here wing feathers in development. And what you see are these just long keratin tubes. These are feathers that are in pin. And there's a few important things to know about feathers in pin. First, the inside of these pins is where the feather is growing. Okay? Right now in these elongate tubes, there's a feather that's all rolled up. There's a rachis and a bunch of barbs rolled up in a little straw. So that's one thing that's important to know. Another thing that's important to know is when feathers are in pin, the pulp tissue is active, meaning there is vascularization at the base of those pins, a blood supply, so that the feather can grow inside of these pins. If you break one of these pins, the bird will bleed because the vascularization is present. You cannot pluck a pin without harming the bird. Not that anyone would ever, I think, try to do that. I don't know why you would do that. But just understand that a young bird with feathers and pin, or an adult bird for that matter, with feathers and pin, if you damage the pins, the bird will bleed. When the feather is completely finished developing, though, that pulp it is all reabsorbed by the bird. As much of it as can be is recycled into the body of the bird, and in that instance, that's the point at which the inferior umbilicus is formed, the hole, and then you can pluck the feather, and there's no blood anymore. So, being cautious with a bird and pin is important. You don't want to break the pins because the pins have a blood supply. Big Fluff here, if you look really closely, there's something pretty cool happening on these pins. These are very long pins because, well, for one, it's putting on big feathers. But if you look, where can we see this best? It's really, there's only a couple of them where you can see it very well. On this one right here. So on this pin right here, if you look, you see it looks like a fan coming out from the tip of that pin. It's obviously not a fan. It's the feather starting to push its way out, and that sheath is degenerating. That sheath is degenerating. That feather is starting to come out of that pin. And you will see birds at different times in molt cycles when they have a lot of pin, and you'll see all the pins, and there'll be all these little tough puffs coming out of the tips of the pins. That's the feathers starting to push their way out of the degenerating keratin sheath, which is really neat to see. Okay. So now we get to your discussion question. So this is your discussion question for the week. The color of feathers is a particularly interesting subject. While most color when we think of color, we think of pigments, right? Pigments create color by absorbing certain wavelengths of light and reflecting other wavelengths of light. And if you're not familiar with how that happens, the book goes into some description of reflectance and absorption in visible light spectrum. But essentially, pigments absorb colors and reflect others. We see the reflected light as it hits our eye, and our eye interprets that particular wavelength as some color that we call orange or green or blue or whatever. Only not all colors are produced by pigments. Some colors are produced by other mechanisms, such as blue actually is a color that is not really produced by pigment. It's produced in other ways. Um, and that's pretty interesting. There are also some structural things that create color effects that are very interesting. So if we look at the bird in the middle here, is a rufous hummingbird. And if we look at the rufous hummingbird, and you look at the male hummingbirds, they have this beautiful gorget. But if you look at a gorget, that's the big patch of feathers here in the chin and throat. If you look at a gorget, there'll be these vibrant colors. You will see they're highly iridescent, and depending on what angle you look at them, they can go from a shiny, beautiful, vibrant color to jet black because of the angle um, that you're looking at them. If you look at uh, most, probably most birds you think of, something like a Baltimore Oriole pictured here, or a Northern Cardinal, most of those birds, you just have normal pigment is the primary source of color. Things like carotenoids and melanin produce those colors. Some birds, like yellow-billed turacos, cetaciform birds, so that's parrots, they have their own unique pigments to create their colors. So like the turaco here has pigments that are specific to turacos that create the greens you see in turacos. It's pretty cool. And then on the bottom right here, you've got, this is a northern saw-whet owl. I actually don't think this is a picture of one of the owls that I worked on. I think I might have got this off the internet, but this is a species that I work with every single fall as they migrate through West Virginia. And they have the UV reflectance in their wings, particularly on the underside of their wings. And you can see that here. And the UV reflectance is really useful for someone like me out in the field because it is useful in aging the birds. Essentially, without belaboring this too much, um, these sequences of regions of high reflectivity and low reflectivity tell you about the age of the feathers, and that lets you age the bird. Um, so there's all these different ways that color is created in different groups of birds, and there's different reasons that those colors are important to different groups of birds. So your assignment is to choose any bird you want to choose. I don't care, whatever you're interested in. You need to describe verbally in text the coloration of the bird so that your readers can understand what colors you're working with. And then you need to investigate what creates the colors in that species. Now, for many species of birds, there's not going to be a specific citation you can find that talks about the pigments or the finer microstructures of the bird that create its color. If you can't find that, the book goes into most of the stuff that you would want to know about color in birds. You can use the book to talk about how the color in your bird is being created. That's totally fine. Obviously, it needs to be in your own words, which means you need to read and understand that section so you can then describe in your own words the colors of your bird and what creates them. The final and very, very most important thing in the discussion, though, is you need to then take the information about color you have and describe why that color is important to that species. For example, what sexual behaviors, if it's a sexual dimorphism thing, are associated with that color? Maybe it's a camouflage thing. Maybe it's something else, right? But those are two really common reasons that birds have the colors they have. They're trying to have sex or they're trying to stay alive, stay hidden. It could be aposematic display. That could be the function of the color. You have to describe in the biology of the bird how color is associated with that biology. So, you pick a bird, describe its color, tell us why the color is the color that it is, what's creating the color, and how that works, and then talk about how the color relates to its biology. That's the discussion for this week. The last thing to talk about, and this is sort of a generic thing, maybe I should put it toward the beginning of the lecture, but this is where it's at, is the different types of feathers in general. Birds have different types of feathers. Most of them you're familiar with, probably. I mean, all the ones on this slide you should be familiar with, um, because we've already talked about these ones. The contour feathers, which are the body contour feathers, and then the flight feathers, the remiges and rectrices. We've already talked about these, so I won't go into them again. There are feathers on birds that you can't really see very well, or you can only see if you have the bird in your hand, for example. But maybe you won't be able to see when the bird just flies up and perches on a tree in front of you. Some birds, such as, for example, herons, so some herons have powder down. And powder down is a special type of feather that is, it's fairly plumulaceous in nature, and it's constantly degrading. The keratin breaks down into a keratin dust. That dust is an important part of the grooming regimen of the bird as it spreads that dust around its feathers, and it adds extra waterproofing to the feathers of the bird. Um, Birds will have bristle feathers around the eyes, the nostrils, and the mouth, and these act like whiskers. And so, what do whiskers do on a cat? Right? When a cat, for example, sticks its head into a dark hole, the whiskers reach out and act as sensory organs in that dark space. So even though the bird can't, or the cat, I'm sorry, we're talking about a cat, the cat can't see around in that particularly dark space, its whiskers, if they bump anything, they give tactile information back to the cat. The same is true. Bristles act like whiskers for birds. And then phyloplumes. Phyloplumes are pretty interesting. They're really, really tiny feathers that are near the bases of some of the rectrices and remiges, and they're different because they're not connected to muscles. They're connected to sensory corpuscles, and so those sensory corpuscles detect really tiny movements in these phyloplumes, and the avian brain is capable of taking all the information from these phyloplumes as they're moving around, subject to the forces of wind, and it synthesizes that information and uses it to control flight. It's pretty cool. And one other sort of type of feather that your book notes that I didn't have in my other list there are after feathers, and after feathers are feathers that are found at the base of other feathers. They are usually smaller than the main feather, and they are insulating, um, much like uh, natal and powder, or natal and adult down. Um, I think I may have accidentally skipped this slide. I realized after, so I got to that slide. Um, Down feathers are insulating feathers that are plumulaceous. That's what they are. They are underneath the contour feathers. Um, they can be defined based on the relative length of the barb to the rachis, the barbs to the rachis. Um, but again, an after feather is just is like a secondary feather growing from the same epidermal collar, which is kind of neat, as the main feather. In the emu, this is the example on the right here, and this is in your book. Um, it's the only bird where the after feather is the same length as the main feather that is growing from that epidermal collar. It's just kind of a neat fact about emu. All right, that was quite a bit. I tried to keep it under an hour, and maybe I over-explained some things, but I know how difficult understanding feather development is because I've taught it before. So hopefully somewhere in that explanation things started to click for you. If not, you can email me and I can describe it in a written way that might be easier to understand. But first, you got to read the book. Um, Obviously, if you have any questions or any, about this stuff, or you want to, talk more about it, you can email me. I'm here for you as always. Uh, have a great day.